Thank you for considering me as a candidate to the Rockefeller University Exceptional Scholars Workshop. It warms my heart to see programs like this that aim to support the development of the next generation of diverse faculty members, and it will be an honor to be selected to this program and have a faculty member like Dr. Jeremy Rock. My name is Alexis Jaramillo Cartagena. I'm a postdoc at the Broad Institute, and I will talk about our work investigating mechanism-dependent phenotypic responses of carbapenem-resistant Enterobacterales. Kira Taylor is the protagonist of a lot of this work. She's a research assistant whom I have the pleasure to mentor. I'm also fortunate to collaborate with clinicians at the Massachusetts General Hospitals and talented computational biologists here at the Broad Institute. Carbapenems like meropenem are a class of beta-lactam antibiotics that comprise some of our antibiotics of last resort to combat a number of multi-drug resistant bacteria. Unfortunately, carbapenem-resistant Enterobacterales, or CRE, are emerging and spreading worldwide, and they are considered by the CDC to be one of our most urgent threats, which cause tens of thousands of hospitalizations and thousands of deaths in the United States every year. Enterobacterales employ multiple mechanisms of carbapenem resistance. They can express carbapenemase enzymes that rapidly degrade carbapenems. And carbapenems usually enter bacteria through membrane proteins known as porins. And bacteria can inactivate these porins in numerous ways. They can completely obliterate the expression of porins, or these porins can also be mutated in a way where they are expressed but prevent carbapenems from entering the cells. And these two mechanisms can also act in concert. Next, I will show a series of plots with our data. This data plots meropenem minimum inhibitory concentrations as a function of inoculum, and we conduct our experiments spanning 4 orders of magnitude in inoculum, and the backgrounds of these plots reflect the clinical breakpoints for meropenem. On dark gray is the resistant breakpoints, on light gray is intermediate, and on white is susceptible. And antimicrobial susceptibility assays are usually conducted in a small inoculum testing range, but by expanding that range, we are beginning to get mechanistic information into the mechanisms of carbapenem resistance. And we observed that strains encoding strong carbapenemases in bacteria like E. coli, Klebsiella serratia, all of them show very strong inoculants effects. By contrast, strains whose carbapenem resistance is driven by porin deficiencies show a nearly constant MIC spanning this broad range of inoculum, and there is also a large number of isolates that encode both mechanisms of resistance and show very highly elevated levels of carbapenem resistance, and we have termed these isolates hyper-CRE. And we show that hyper-CRE are the result of the synergistic action between these two mechanisms of resistance by taking a carbapenemase expressing isolate, knocking out its major porin using CRISPR-Cas gene editing, and showing that this strain became a hyper-CRE. Similarly, we took a porin-deficient strain, transformed a carbapenemase, and showed that that strain became a hyper-CRE. Next, we ask if all the structurally distinct carbapenemases impart an inoculant effect. To answer this question, I clone all major carbapenemases into plasmids. I transformed those plasmids into susceptible strains of E. coli and showed that all of them exhibit carbapenem resistance and an inoculant effect. One of the most important implications of this work is that it has shown that a large number of CRE can test as susceptible in antimicrobial susceptibility assays, which poses a threat for the treatment of patients. Lastly, we investigated an isolate that encoded wild-type porins but displayed very large levels of resistance. I performed in vitro evolution with this strain and obtained a series of derivatives that became more susceptible to meropenem. Each of these derivatives had a different mutation in a transcription factor. And we have since realized that this factor also increases resistance to other classes of antibiotics like quinolones and tetracyclines. And continuing to characterize this factor might propel the launch of my independent research career. I want to thank my PI, Dr. Robi Bhattacharya. I was the first postdoc to join his lab, which has accelerated my scientific growth. And once again, the protagonist of this work has been Kira Taylor, an outstanding research assistant. And thank you for your consideration.